Hi there, Mr. Dahl here with you, and today we're going to recap what we learned about the cell cycle, most specifically interphase. So when we think about the cell cycle, what's the whole goal? Well, we're starting off with one cell. We want to make copies of our genetic material and pass it on to two new daughter cells. Now keep in mind this cell cycle that we're going to be talking about is for eukaryotes, meaning these cells have a nucleus. Now when I think about the cell cycle, I think about it as a circle. Two reasons. One being that it is a circle when we show a diagram of it, and I think about it because it's the circle of cellular life, just like the circle of life in Lion King. So there's my Lion King reference for the day. So here you go. Here's the eukaryotic cell cycle. This is our diagram. We're going to break it down into its component pieces and talk about each piece. So the first phase of the cell cycle, we have G1 or gap 1 phase. The only thing you need to know about this phase is that this is where a lot of the growth occurs. This is the adolescence. The cell is growing up, getting larger, and maturing so we can make another cell go through the cycle again. If it doesn't go through this growth, or if it, gets stuck in this growth phase, we're going to go to another phase over here, or we can go through and proceed along the cell cycle. If we get stuck over here, we call that the G0 phase. This is a phase of no cell division. The cell is stuck here, won't move to the next stage, won't divide. An example of this is nerve cells. Nerve cells can't divide to repair damage to them, can't replace themselves. So this is why if you sever your spinal column, damage those nerves, you can be paralyzed for the rest of your life. These cells are stuck in G0, they're not going to divide to replace themselves, you're not going to be able to fix that spinal cord. So if our cells are not stuck in this G0 phase and they move on to the next phase, they go to the S phase. S for synthesis. So what's occurring here? Well, in our parent cell, we have our chromosomes here, but we only have one set of our genetic material. In this S phase, we're going to make it into two new sets because we need a set for each one of our daughter cells. So we're going to split the double helix, split the ladder, and make a copy of the new side to make two new copies of our DNA of all of our chromosomes, so that eventually when we go through mitosis and cytokinesis, each daughter cell is going to get a whole set of DNA. So after this occurs, we move on to the G2 or gap 2 phase. Now there's three things you're going to want to remember about this phase. First thing, it's another growth phase, that's why it's called G2, doing a final growth, getting large enough to divide and make two new cells. What else is happening here? Well, we're also making copies of all of our organelles, so that each daughter cell can also get a complete copy of organelles to carry out the processes that it needs to live. And finally, right here, we have what's called a checkpoint. Now, this is going to be a checkpoint to determine whether the cell has made the right copies of DNA, there's no mutations, no problems with the cell, so that we don't make copies of a bad cell. We only make copies of cells that we want to make copies of. So we're looking for mutations in the DNA, we're looking for problems with organelles, problems with the cell, and they're only going to proceed through to this next step right here if everything's okay. If everything is okay, we finally go to mitosis and cytokinesis. Right here, this last arrow, where the cell is actually reproducing, actually dividing. We're actually taking that one cell, and remember we've got two copies of the DNA, and we're making two new cells, each with their own copy of everything. But mitosis and cytokinesis are a topic for another day, another lesson, and that's what we'll be moving into next time. So just to recap, this whole portion right here is what we're concerned with, not mitosis. We're concerned with interphase, the rest of the cell cycle, when it's not actually dividing. Hope that helps. I look forward to talking with you about mitosis and cytokinesis next time.